Hi, everybody. Welcome to Young Academy of India's YSL series, that is Young Science Leader series. And today with us, we have Padmini Ramachandran, my good friend. And Padmini is working for one of the prestigious policy think tank, USFDA. That is Food and Drug Administration of the federal government of the US. And she's over there as a biologist, and involved with the food and drug testing. Padmini is expert in using next-gen sequencing, the DNA sequencing technologies to source track food outbreaks. So that is what I think she will share a little bit about her experience today working for the USFDA. And Padmini is recipient of several awards and accolades within FDA for expertly using metagenomics inbuilt environment and agriculture setting for tracking food outbreaks. Very interesting, and especially on this metagenomics part and next gen, how you are using for tracking the trails of the outbreaks is exciting. I would like to know a bit more about how you do. And yeah, so welcome, madam, for our YSL series. So I'm really happy to have you here with us, and especially this is a Young Academy of India. Is an academy where most are young, aspirants, mostly the PhD students and young professionals across India. So yeah, so we are really waiting to hear your story. So it's more like an interview fashion. And today with me, the co-moderator of this session is Nitya. And Nitya has already moderated, not moderated, she did a translation of The Curiosity in Telugu a few months back. One of the episodes she did it fantastically. And Nitya Kriti is, she is a BSc final semester student at Government Degree College in a township, which I forgot the name of the township, I hope you will say, near Hyderabad in Telangana, and where she is doing BSc in biotechnology. Over to you, Nitya.
Thank you, sir. Good morning, everyone. My name is Nitya Kriti, and I'm very glad to have this position here. So, talking about my previous project in YSL, I had a chance to work on frequent urinary tract infections in women from Amity University by Nitoma ma'am. And this was an opportunity that I found from the Felix Bass channel. And I hope many of the students has got opportunity to work on such real-time projects. And I would request everyone to take up such real-time projects, especially for the life sciences student, even this year also. And today we are very glad to have Padmini Ramachandran ma'am here. Ma'am, good morning.
Good morning, guys. How are you guys doing?
Yeah, all good, ma'am. Thank you for asking. Ma'am, could you please tell us your story and a bit about yourself, your family, and your early childhood?
Sure. First, thank you for having me, Felix, and thank you, Nitya. I did prepare some slides, so I'm going to share the slide. I hope.
Yes, ma'am, it is visible.
All right. Okay. So thank you. Like I said, thank you for having me. And like Felix mentioned, I work for the Food and Drug Administration in the US. And my main research idea is applying metagenomic methods to food safety. Before we move on, I have to have this disclaimer. Whatever that I'm sharing today are my personal views and opinions, and they are not of my employer, who's the Food and Drug Administration. I just want to make that clear. All right. So thank you, Nitya, for starting me off with the most easiest and wonderful question. So I know that family is the most important thing, and they are the driving force behind us, right? So my parents are from very, very humble background, and my dad worked for the government, and then we are four kids. So in India, as you know, from the childhood, like if you want the kids to go to a private school, you have to invest money in education. So my dad, he wanted to have his kids to have the best education. So he invested in private school for all four kids. And then my two elder sisters, they are married and they are homemaker. And I'm the third one. And my brother. So we both are the working kids for my parents right now. So I got married in 2005. No, sorry, 2006. I think. No, 2005. 2005. And then I came to the US. And one thing that I was very clear was to keep working in science, just because I did my bachelor's in KRMMC Chennai, a college in Chennai, and then I did my master's in biotechnology along with Felix and several other really, really good-natured students in University of Madras. So in that, so many of my classmates went on and did their PhD. One is Felix, and you have met Shaktivel in YSL before a couple of weeks ago, and Ramesh, Sevbel, Dinakaran, they all went on to do their PhDs. And I was like, oh, okay, so PhD is the right correct next step. So I really wanted to do it. But then as a girl, and I'm the third girl in the family, and my parents were itching to get their duties done. So they got me married, and mine was an arranged marriage. To a very, very traditional family, and I am also from a very traditional South Indian family. In 2005, so I came to the US in 2005. And one thing was my husband really encouraged, you just have to go, explore. And if you want to do in science, you just have to do it. So I was pretty much scared, and this because it's the first time in the US, and I am really new, and I have not explored a lot of things. But my husband really encouraged me. And one thing that was pretty challenging was the work visa. So no company was ready to sponsor H-1B, especially for the science and without a lot of work experience. So I, at that time, had in touch with one of our junior from Madras University who did Masters in Biotechnology. Her name is Vidya. So she was working for an university in New Jersey. So I got in touch with her through email, and then she shared her phone number with me. And then she said that universities are the only ones who will sponsor H-1B because they are grant funded. So, okay, so what do you mean by grant funded? So every year the grant gets renewed, and they ask for people to help in the lab, like a lab technician. And then every year the H-1B is renewed. If the grant is for 3 years, then you do have the H-1B for 3 years, and then you can work in that department. So I was like, okay. So then I started applying. The area where I live in DC suburbs, so Washington, DC suburbs. So there were 2 or 3 universities, and one of the best university was Johns Hopkins University, or best university in science, I should say. So I started applying through the university website, and luckily there was a grant funding available in Department of Emergency Medicine along with the Department of Infectious Diseases. And then they were like, oh, yes, and we need somebody who can lead this study where we are doing a rapid diagnostic assay development for sepsis. So especially bacterial sepsis is pretty intense in the sense that the bacteria gets into the bloodstream, and within 6 to 8 hours, you develop severe symptoms like extremely high fever. And the bacteria, since it enters into your bloodstream, it goes into several organs, and then there is organ failure, and eventually the patient dies. So they wanted to have a rapid diagnostic assay, which was funded by Department of Homeland Security. at that time, where we have to develop a diagnostic assay less than 4 hours, where we can detect the bacteria in the bloodstream using PCR, polymerase chain reaction, less than 4 hours. So that was the purpose of the grant. And they wanted, they had three PIs, three principal investigators for that grant. And they wanted somebody in the lab who can think and problem solve as it goes, because the principal investigators are actually doctors. They are medical doctors, so they will be in Department of Emergency Medicine and Department of Infectious Diseases, where they will be looking at patients and they will be enrolling patients in the study as they come in. So their job is that, and their job is to have the correct questions, like, yes, how to develop a diagnostic assay rapidly that can detect sepsis in less than 4 hours. But they wanted somebody in the lab who will think on their feet and fix problems and carry out the research. And they really liked me for some reason. Thinking back now, like 15 years back, I don't think I was this experienced, or I was pretty much naive. And I was right out of college, pretty new to the country. And I was like, I can do this. And it was the best opportunity of my life, because I learned so much, how to think on my feet, how to design experiments, and what are the correct questions to ask. And if those questions aren't answered, what would be the, like, so then if the questions aren't answered, what is the next correct question that will get the bigger overarching goal answered. So that pretty much set in my science career path the experience at Hopkins. And then eventually we did publish a couple of papers, and the Department of Homeland Security took the rapid diagnostic assay to ELISA, and they were very impressed. But the grant wasn't renewed, so I had to start looking outside Hopkins. In the meantime, because of the contribution in science, I had gotten my employment card within the US, where I can work for any employer. Then luckily, FDA had a research fellow funding at the master's level, which is very unusual for US FDA, where they would have a research fellow funding at the master's level. I did apply, and because of my experience at Hopkins, but I had developed rapid assays using PCR and some of the sequencing technique available at that time, I got the research fellow opportunity at FDA. And then eventually I was enrolled as a biologist in FDA, because one of the studies that I pioneered was using next generation DNA sequencing technologies of the microbes that would really help in source tracking food outbreaks pretty early on, so which will help the FDA. So my inspiration is a combination of factors, as you can see. Definitely my parents, definitely my dad, and then my husband, and definitely my mother-in-law. So like I said, it's very tough for women to work and to become a very successful career woman. And especially in science, it's not that you have a 9 to 5 job. You have so many research experiments that would go overnight, that would need like several long hours of planning, designing, coordinating, and everything. And my mother-in-law was there from the beginning to support me and to help me with my kids, to help me with my day-to-day chores, because leading a family is always a 2% job. So she did help me out, and she was encouraging me throughout my career, even today. If I have to travel, because my job takes in travel, where I have to train all the FDA counterparts and countries, the FDA in other countries, for this genome sequencing technology to apply in food source outbreaks. So she is a pioneer, and she's one of my inspiration, and definitely my kids. Every day they always make me a better person. So I work for them. And one of the pictures that you see here is the first generation Da Vinci surgical robot, which can do surgery on its own. When I came out of Hopkins, this Da Vinci robot, I got a few opportunity to see this Da Vinci perform surgery live, and the precision medicine was the first it hit me, like, how important precision medicine is, how important precision diagnostics is, how important digging deep and looking deep into this molecular level and understanding the gene functions. And more than the phenotype, the genotype correspond 90% of the phenotype. So how important it is to minutely understand it in detail for us to be a better scientist. So this is one of my inspirations also, the Da Vinci robot. So these are definitely set my career path here. And I would definitely fail if I don't acknowledge my mentor in FDA. Her name is Dr. Andrea Artisan. So she's one of the pioneers in metagenomics. I would definitely explain to you guys what metagenomics is soon, but she has so many ideas. She's the most creative person I know. When you are in science, in the field of science, being creative is one of the challenges. Like, I'm a scientist, I know, so every day, like, it is very tough for me to think beyond a certain box. So she is one of the most creative person that I have ever met, and she is my inspiration every single day. And she from the beginning, she has helped me design so many schemes for next generation sequencing technology, the gut probe arrays, and working on a lot of bioinformatics pipeline that would answer the science question in the lab. Because you have a great divide in scientists, like who are bioinformaticists, who are programmers who can develop algorithms, and you have us people like who are in the lab who can think only science, but you don't have person who are interconnected, interrelated, finding that niche where the lab person has to think bioinformatically and annotate those data sets and develop algorithms, and they also have to design experiments. Those kind of niches was identified for me because of Dr. Andrea Artisan. So she is one of my inspiration, and she is still within FDA. She has moved on from Center for Food Safety to Center for Veterinary Medicine, where she is looking into a lot of plant poisoning for the animals, like generally livestock. So her passion is using metagenomics for that, but she was a true inspiration for me. Yeah. So that's just to answer your question.
Yeah, I truly understand that it was not a cakewalk journey for you, ma'am. And I'm very much sure this story would be inspiring for many budding undergraduates, graduates, and upcoming scientists also. Felixa, do you have any word to say?
Yeah, I mean, I was just looking at those pics, especially, you still have the pics from the embassy, and amazing the journey, it's a really inspiring, Padmini. And I'm just wondering, do you have any mentors when you were young? You said about, of course, your parents were one of the main influence, and also, of course, your in-laws. So is there any mentor or books that you read as young that it actually immensely helped you that you would like to share?
Yes, definitely. Like I was a very typical CBSE kid, where I was introduced to Sidney Sheldon and all that. But eventually, as I grew up, like when I went to high school, there are a lot of science fiction where they use DNA. On forensics and all those novels, like I was like, oh my god, like, DNA is so tiny, you don't see it. It's not like blood where you swab, like, you don't see it, but within the blood you have this very tiny coded double-stranded structure right there that's going to tell everything. So I was pretty inspired by those novels. But my science teacher in my high school, my 11th standard and the 12th standard high school, she was she was fierce woman. Like, she did her bachelor's in zoology. She did her master's in zoology in Meenakshi College for Women, which is a very, very known college in Chennai, and it's very tough to get into. And her goal was to become a doctor. And she always used to tell me, like, okay, so for forward cast, like, getting into a medicine field is pretty tough. Like, you have to have a 99.9999% on your high school and your medical entrance exam for you to get that. So I wasn't that smart in high school, or I was not that driven in high school. I am from a forward cast, and I didn't have that score. But she would always say, like, who would have doctors, all these scientists who develop these diagnoses, these treatments, these medicines, like, who help them answer those questions. Those are the one. And she was a fierce zoology teacher. And at that time, I didn't know microbiology. I didn't know microbiology existed. It was like 20 years ago. I graduated from my BSc in 2000. So 21 years ago, microbiology in Chennai was not that heard of. So I was aiming for a zoology coursework. But then she was like, oh, my neighbor, right next door neighbor, her daughter does bachelor's in microbiology. And they are the first batch in their bachelor's college. And I was pretty hesitant because I will be a second outgoing batch from that college if I had taken microbiology. I talked to her, like, I used to go to her house every day evening because we had these tuition that you take, right? So you go math tuition, you have biology tuition. Even though biology, you don't need tuition, you have to do it. Like, that's how the world operated. So I went, and then she was like, oh, you know what? You should definitely talk to her. And those days, you pretty much followed the path like a herd. Oh, people does IT, people does computer science, people does math. Oh, this is the college. Oh, someone told me this college is nice, so you apply for that. You generally don't sit and talk to the person who's actually taking the course, which was a little bit new to me. And I was like, you want me to talk to her? And she was like, yes, you should ask her questions. And that's the first time I was like, yes, I can ask whatever questions I have. I don't have to ask whatever question my parents have or like my neighbor has. So let me go and talk to her about what this course is about, like, what does it deal with? And then she was like, she was pretty hesitant because the lab setup and everything was in that college. Since they are the first batch, it wasn't that great in the sense it they didn't have a lot of equipments or anything to work with. So she was like, oh, if we want to run a gel, we are like 22 students in the class, and only the teacher runs that agarose gel, and not all 22 get that experience. I was like, oh, running a gel and looking at DNA. And I was fascinated. I don't care if I don't get to run a gel by myself. The teacher is running it. So I get to watch the DNA go from the top to the bottom, which was super exciting, like 20 years ago or 23 years ago when I was in high school. And I was like, okay, I'm not doing zoology. I'm definitely doing microbiology. And I still owe it to my high school biology teacher, mentor. Like, she was a true mentor in the sense that now that I think about it, at that time, I didn't know mentor, mentee. But now that I think about it, I know more. how much of a role that she played. If she hadn't told me, go talk to my neighbor, I would be like, oh, everyone is applying for zoology and you did zoology, I would just do zoology. I wouldn't have asked the right questions. So she encouraged me to ask the right questions. I asked the right questions, and it's all that day, that evening, that tuition that changed my life, which seems very, very tiny in the path, but that makes a profound difference.
Yeah, very exciting. It's like black swan event, the chance events really play a tremendous role in shaping our entire life and career, isn't it? Over to Kriti. Do you have any question?
Yes, let me admit one thing, ma'am. You're not only a great scientist, but also a good storyteller because I could imagine each and every part of your story.
Thank you. That's much needed, in science, because scientists in general, at least here, the opinion is they're so boring because they express only data points. Like they talk about data, they say that, oh, I identified this, I identified that, but they don't tell you how did they arrive to that question? Why is that question very important? And when you find that data point, why is it very, very useful? And that's going to change the science career path or the next step moving forward. So, and also another very important thing in science is to write. If you're not a good writer, that's I'm not discouraging, but focus on your writing, practice every single day, not science writing. Just write the events of the day as a story. And I am positive, like after a week or so, when you go back and read the week before story, it would have definitely improved. So you need storytelling to be a better scientist or to be recognized as a better scientist, I guess. Thank you for bringing that
Yeah,
up. That's a really good,
yeah.
valid point.
Yes, ma'am. And ma'am, why did you choose a career in basic sciences? And do you think after all these years, do you consider the initial decision was rewarding or unsatisfying?
That's a great question. Yes, my initial decision, I wouldn't change even one single thing that I did. Because whatever that I did is I am today has put me in this position today, working for FDA. So I was telling Felix before that my husband is an architect and he designs buildings and town planning and all that stuff. His work is pretty rewarding because whatever that he designs, like in 5 or 6 years, he sees that building right there, and it's very rewarding. But whereas in science, you do a research, you publish a paper, and for the science to be adapted practically in everyday aspect of life, it takes years, years. But with FDA, the thing is science get translated to policy, policy get translated to regulation. Regulation means people have to follow. And especially in the outbreaks, food outbreaks that I am in, the source tracking I'm in, if FDA advises people not to use this water or use this particular type of testing, and then use the water for agriculture, like all those policies and regulation put forth by FDA comes from scientists within FDA who are working towards that. So when I see that my science or my research question or the research question of the public gets answered by me and then gets translated to policy, gets translated to regulation, then adapted by the public as means of public health, it's so rewarding. So whatever that I did initially, I wouldn't change anything because every single step that I took, doing a microbiology, doing masters in biotechnology, and then coming here was not my decision. It's that's just fate. That's the only thing that I didn't take any initiative towards it. But as soon as I came in, everyone was discouraging in the sense that they were all IT. They know how to get a visa for an IT field, but they didn't know much about science. But my husband, my in-laws, and my parents, they all encouraged. So I got in touch with so many people And then they showed me a path. And then I went to Hopkins. Hopkins is one of the leading medical universities. And I got to work with some great minds. And if I hadn't done the initial decision to do basic sciences, I would not have met my Hopkins PI. I would not have known like how it is to work in a very intense environment. Emergency medicine is pretty intense environment because you have people coming in who you have no idea what they have, but they will be dying. So you have to think on your feet. And then the doctors, the PIs, the principal investigators that I work with, like I said, sepsis, it kills a patient in 8 hours. You have to figure out what bacteria causes that sepsis, and they have to start the treatment immediately, or else they lose the patient. So nothing is nothing comes easy. Like you have to be really quick. In science, being quick is not heard of, but I learned all that, and I wouldn't have be here like if not for my initial this one. So I'm pretty happy. Yes, it is rewarding.
It's very reassuring and encouraging words, Padmini, because there are a lot of trends these days that the people, who is after the scopes and trendy fields and professional fields, professional courses like engineering and medicine, completely forget. And, in India, the situation very well, that the really motivated and very brilliant minds are now more and more not choosing the basic sciences. They are going for the engineering, of course, IIT, JEE, they try to crack, or MBBS, and completely forget. So we end up with only the mediocre in the basic sciences. So I think that is actually happening everywhere as well. But in the West, the trend is kind of less. And I'm really happy that, after listening to your humble origin, and you did a fantastic work on, you choose a career in basic sciences, and it's highly rewarding. Now that you're the top of world, I can say. Working for the US FDA is definitely, it's a huge privilege. And more than that, you actually won multiple rewards in your workplace, as the best employee awards. How did you make it, Padmini? What is your experience in US FDA over these years, almost a decade? And especially, I would like to know a little bit about your productivity tips that you learned, you picked up from the FDA.
Yes, definitely. That's a great question, Felix. I'm so happy that you asked that question, because one of the things with FDA is it is also a high functioning environment. When I say high functioning, the public health is at stake and the outbreaks are happening real time. And if the outbreaks are happening real time, you have to stop the outbreak. You have to detect what organism is causing the outbreak. So then you advise the farmers, the growers, the food manufacturing, the inbuilt environment, storage facilities, and everything on where what went wrong. And next, you have to prevent it from happening again. You don't want the consistent outbreak happening again and again and again. So it's a very high functioning environment if you're not organized, and you also have to think ahead, because all the if previous science has worked, or the way that we do farming, but the way that we do agriculture has changed quite a bit, and so many of the old techniques doesn't, I wouldn't say help us anymore, but it doesn't work well in the high functioning environment. So in that case, you have to come up with a lot of next generation ideas and quick thinking. So you have to stay organized. One thing is the FDA has trained me is that even in a high functioning environment, since we are a regulation industry, if we go after a particular company or anything at all, we should back our story with very strong science. If we don't, we are in trouble. So in that case, how to do strong science, how to do reproducible science, how to have repetitively the same result again and again. So those takes precision. So that I learned extremely well in FDA, because I would be held accountable tomorrow for putting forth any of this data. It's not that I'm going to publish it, 10 people are going to read it, and that's it. No, these are getting adapted towards public health policies. So I have to be very precise, and I have to have reproducible results, and I have to back up with a very good science story, so that when these policy becomes regulation and the public has to adapt it, they should understand what they are adapting this for. So if I say don't wash the chicken, then the bacteria in the chicken is going to spread all over the sink, and then you're spreading the bacteria everywhere in a kitchen environment, and then you're eventually contaminating the other food source also. Then I have to back that story, that very simple cleaning technique, with a strong science background, but to the public it has to make sense. So it's a very high functioning environment. It's a high precision environment where I have to have every single step recorded well. And the third is really practically science, like people should be able to practice it. When I go and tell a grower or a farmer to avoid lettuce outbreaks or to avoid salmonella outbreaks in red onions, do this, do that, and they're like, who are you? Like, I've been doing this farming for so long, and if you come and tell me this, not to water these plants or test my water before I do this, or like, check my sprouts, like the seeds, to do this, like I should be. So that's one thing that I learned from FDA, because when science gets translated to common people as a regulation, how should I present it? And how should I do science to begin with? So I have to have precision science, and I have to have reproducibility, and I have to have repeatability. So how do I do that? And statistically, how do I back that data up? So those are really good scientist-oriented goals anyone should have. So I think that I learned. And another thing that I learned is that no matter what you do, government is at blame. so so that's also something that you should keep in mind. So it might not be rewarding at times. So that's another thing.
Yeah, very interesting. Yeah, you emphasized on the empirical and evidence-based policy making, and yeah, of course, a rationality of the science. It's very hard to convince the traditional wisdom, especially it will lead to something called cognitive dissonance. You have a hard-held belief, and if the science say that is not right, for example, you correctly said about the washing chicken. I told many of my friends, do not wash chicken, but they will not be convinced. The chicken that you buy from the market is completely, I mean, it's full of, the dirt. That is what the common wisdom is. And you should really wash nicely in your sink, and then you cook it. But in that process, actually, you're contaminating the entire environment, right? You just emphasized on that point. Yeah. So there is always a clash between what is deeply in our belief system and what we have been following as part of the tradition, and what the science say, right? So that conflict is, I mean, how to look at that conflict. I think that's a very important point that you emphasized on on your answer.
Yes.
Nitya, you have any question?
Yes, sir. I feel this is a good question because I strongly believe that this is something much needed in this pandemic days. And the question I have is, what are the qualities of a good science leader in your opinion? And do you have any role models when it comes to science leadership, especially?
Thank you for asking that question, India. That's also a great question. So my role model in science is definitely Dr. Andrea Artisan, who I have worked very closely with. And second thing is, any woman, not only in science, are my role model, because they have to manage kids, the house, and the job. So that's in India. Like when I grew up in India, I never really realized the strong support system that we have. If women wants to go to work, as long as the husband says yes and the in-law says yes, they can, because they can leave the kids with the grandparents, right? So you have full confidence in the grandparents taking care of the kids. And if not for your in-laws, then you can move near to your parents and then they can take it. I'm not saying everyone has that advantage, but most of them do. But in the US, like you don't have that. Like the daycare, sending the kids to daycare was a very new concept for me. And in my family, no one has work. Like I'm the first woman who had to come and work. And my mom was like, "Oh my God, you're leaving your kid in daycare." And I was like, I'm not sending him to a circus. Like I'm sending him to a daycare where they are going to take care of the kids. And my mother-in-law at that time was like, that's okay. If you want to send the kids to daycare and if you want to keep working, do it. So she's my first role model. And second role model, like I said, is Dr. Andrea Artisan, who is the best creative thinker that you can say. Like, she is very creative and she approaches science in a very, very, very practical way. So she is very practical in the sense that, all right, we did this experiment, it didn't work, and we have spent 6 months on it. The question is not answered. All right, if the question is not, it's not answered, we should ask the correct question. So change the question, like, those kind of thing. She taught me so much, and she is a really good role model. And like I said, any working women with kids and family where they have to cook 3 times a day, yes, they are.
Yes, Padmini, very interesting. And you have been working with the next gen sequencing. Yeah. So I would like to know a little bit about what you exactly you do there. So basically the barcoding, you say that now it has become like a powerhouse in forensics, isn't it? Be it in tracking disease like, the disease outbreak, the food outbreaks, or even the disease like COVID-19 or the medical forensics, deliberately infecting. I was reading one story about Florida dentist deliberate infecting its patients with the AIDS, HIV virus of a decade ago, traced by the DNA barcoding. So do you have any interesting, memorable instances or anecdotes over the years working with FDA on tracking the disease outbreaks, the foodborne diseases? Yeah.
That's a great question. I have some slides prepared just to tell you guys some basics of what the genomics is and how we source track the outbreak. So I'm going to share my screen. All right.
Yes, ma'am.
All right, hold on. I'm unable to move on to the next slide. 1 second. Yep. Right. So one of the things that Felix asked a great question on how the FDA or how do you use genomics or how the DNA barcoding helps in source tracking, detecting outbreaks, or the personal experience that I have in this. So one of the things that I do is something called as metagenomic sequencing. What do I mean by metagenomic sequencing? I take the whole commodity or either the ice cream or lettuce or red onions or peaches or peach leaves or anything at all. And then we extract the total DNA that is there. So I will have plant DNA, microbial DNA, viral DNA, anything. So I would have a DNA extraction bias there, but I would take anything, any commodity, the total DNA of a commodity, and then I just do sequencing. So we do something called as long read sequencing and we have short read sequencing. Those are two different technologies that we use to understand the DNA sequences in the sample. So right now, I have a mixture of DNA of my food, all the microbes in there, and anything that's in there. So from this mixture, how am I going to weed out the microbe that I'm looking for? Anything that I'm looking for. So I query all these genome sequences against a database, a bacterial sequence database or a viral sequence database or a fungal sequence database, or an eukaryotic sequence database that uses chloroplast genome sequences or mitochondrial genome sequences. So then I say that this is the organism that's present depending on the answers I get back. So in this case, what I'm doing is I am identifying the source of contamination or anything like that causes illness within 28 to 32 days. Actually, I know people say that, like, it takes days, but the illness itself in the public health, it's identified at like 23 days. That's when it starts getting identified. So from the day of the illness is identified, within 4 days, we can do the source tracking. One of the really important information that you have to understand is that the basic definition of metagenomics is the analysis of genomic DNA from a whole community. So you extract the DNA from the entire community, you sequence them, and then you're looking at the sequence. So what I'm trying to do here is that we wanted to enrich, like traditional microbiology. What they would do is they get the commodity, the food commodity, they enrich it in a bacterial media to identify the bacterial source of contamination. They incubate the bacterial media for 48 hours, and then they plate it, they isolate a colony, and from single colony, they extract the DNA, and then they sequence them, and then they cluster them. But what we are doing is that we are saying you don't have to wait for every single colony to come there and then take DNA only from one type of bacteria. No, you can analyze it as a community from here. The 28-hour time point enrichment, we take it, we directly extract the DNA from this 28-hour, and we sequence them, and then we cluster them. So when we cluster them, you look at the phylogeny of the bacteria based on the genome sequence. Then you place them with the previous outbreak and then tell them, oh my God, all right, so this is a particular outbreak that has happened in the past. This is the organism, this is the cluster, this is the, within several SNP differences, you have this here. And this is one of our very recently published paper where we have used metagenomics to identify the Listeria organism from an ice cream outbreak. So this was pretty rewarding because FDA for so long were relying on a single colony and a single bacterial genome to identify the outbreak. But now we actually preponed it, like we made it pretty rapid by saying that we can identify the source of the contamination and the organism that's causing this 4 days prior by using this metagenomics. So coming back to Felix's question, now that we understand what we do, coming back to Felix's question, these DNA barcoding, like where you are identifying certain markers within the DNA from your sequences, that actually helps in identifying the source. So even for SARS-CoV-2, right now you can go to several websites where they do have the phylogeny displayed, saying that the Chinese SARS-CoV-2 or the SARS-CoV-2 strains from India, or SARS-CoV-2 strains from the US, SARS-CoV-2 strains from the Netherlands, has all been sequenced, their genomes has been sequenced, and when you put them in two SNP differences, one SNP differences, they all cluster together. That means that once they have entered a stream, they have mutated and they have started clustering. So this is very essential. This is how you trace the outbreaks. This is how you eventually find out what is the organism that's there and which cluster it is coming from. So the DNA barcoding for us, it was very source when we published this in 2019, I think, where we did use it for a real-time outbreak to source track the listeria, like ice cream, bluebell ice cream, and listeria. So I just want to make clear again, these are the opinions of me and myself only, not of my employer. And I have to have the disclaimer. But these are all open source science article. You can definitely go and read. But again, anything that I say today is of my personal opinion and not of my employer.
Yeah, really exciting. It's something like, you save a lot of time from the normal barcoding to the metagenomics, because it's something like a heuristic, one stop, so much of the information coming up, isn't it? Over to Nitya.
Yes, sir. Ma'am, some believe that phylogeny based on a gene or few barcodes have now become old-fashioned, and genetics has been eclipsed by genomics. Do you agree with this? And is phylogenomics more practical in tracking outbreaks?
Yes. Yes. So there are two parts to your question. The first part is that people believe that some of the barcoding is yes and no. For certain aspects, we recently published a paper where echinacea people use in herbal tea. So historically, for the plants, they have relied on 7 or 8 marker genes from the chloroplast genome, like RBCL or MATKB gene, where they will use that as a marker to identify the adulteration in star anise, echinacea, which are the common adulteration, where you look exactly like echinacea or star anise, but it's not that. So historically people have relied on 2 or 3 genes to identify the particular plant. But as people are getting really smart, the adulteration industry is getting really smart. The economic adulteration is pretty high. So in that case, relying on only certain markers is not helpful anymore, because even though, especially with echinacea, it's very hard to distinguish echinacea based on just the 7 marker that has been historically used. So for echinacea, you need to look at whole chloroplast genome of echinacea to identify the differences within echinacea species. So to answer your question, for certain aspects, it does work. The old DNA barcoding works, especially in seafood. FDA has historically regulation in economic adulteration of seafoods. People will think that it's salmon, but it might not be. People will think that it's tuna, it might not be. So historically, what FDA has done is that use certain barcoding genes just to identify what type of fish it is. And it is, even now it's used in regulation. That being said, it doesn't work in tuna. So in tuna, when you have like ground tuna, the barcoding does not work. So in that case, you need the entire mitochondrial genome to tell you whether it's tuna or not. So DNA barcoding, yes, it works. It still works for certain cases. It's still the cheapest method that's available. So whatever cases that it works, people use it, and it has to be used. And if it doesn't work, then you move on to the metagenomics, where you use the entire mitochondrial genome to distinguish and to identify, or to use the entire chloroplast genome to distinguish or identify. So I know there are people who say that it doesn't work. Yes, it doesn't work in certain cases, but it does work in a lot of cases, so still believe that. Next is the phylogeny. Phylogenomics, I just shared the slide. Yes, we have successfully used that in tracing outbreak, and we still successfully use live outbreak recalls. It's called as genome tracker. When you guys get a chance, please look up FDA genome tracker. They do live clustering. People, like whenever the outbreak has happened, all the state labs, they get the food products, they sequence it, they upload the sequences on NCBI live stream, and it get clustered live stream, and you can check the phylogeny, all the SNP-based analysis live stream. So please check on FDA genome tracker. So I am part of the genome tracker where we do metagenomics.
Great to know you're part of that team. Fantastic, Padmini. So
Yeah,
because you're working in this.
very informative, ma'am, and very exciting to know what's happening around the world.
Thank you.
Yeah, so you're working in this field for quite some time, the DNA barcoding and phylogeny. So I'm just wondering, what is your take on this, the Chinese hoax, which is now coming everywhere? I mean, people are believing in this hoax, and do you think it's hoax or not? So the what I'm referring the Chinese hoax is that, some people believe that this SARS-CoV-2 is originated from a lab instead from the naturally from the horseshoe bats. So what is your take on this issue?
Um, Again, these are my personal views and opinions and not my employers. Um, I I don't. So based on the genome sequences that's been published, and based on the phylogenomics that's been published, and based on the clusters that we have seen so far, just based on the science alone, I don't think it's from the lab. It would be that originated. But I don't have proof otherwise to tell me, science-based proof otherwise to tell me it's not from the lab either, where people haven't created this in the lab and they have not published genome sequences from that. But based on the available genome sequences, the science and the clusters in which they are placed, I don't think it's from a lab. So that in itself answers your previous question, Felix, like how much do you trust science? And it's very tough for people to trust science, and it gets you only so far. And yes, I understand that, but I have trusted science so far, and I will keep trusting science till the day I die. So for me, the evidence is not otherwise, so I don't think it's from the lab.
Yeah, fantastic. Yeah, I'm glad to know that you trust science. So well, it's immaterial trust or not, because the science deals with objective reality, and still people, I mean, they spread the fake news and conspiracy theory. It says that what I heard is like, when the truth takes, it goes on the snail's pace, then at that time the fake news would have been crossed the other side of the planet Earth. Fake news and conspiracy spread so fast than the truth these days, isn't it? So the social media is full of fake news and alternative facts. Yeah. Nitya, you have any question? We are ending the show. It's already it's almost 1 hour. Yeah,
Oh my
it's
God, I've
Yeah,
talked a lot.
enjoying this
Yeah,
talk.
yeah, 1 more minute.
yeah. Just a final question from my side, ma'am. For a fresh PhD from India who aims to get a job in FDA like you, what are your recommendations?
Definitely do a postdoc here, because you would understand how research lab operates. It's very different from what I have seen in India. I'm not saying anything negative. I'm saying that it's just that the thinking itself is a little bit different. And uh, and the way that the resources that are available. So you would really learn how to use the resources to the fullest so that you can become successful. So directly, instead of applying for jobs, I would definitely do a postdoc here, understand the lab setting, and understand whether research is for you or industry is for you. So when you come here and give it some time, you might be able to understand that. See, if industry is for you, then look for jobs in industry. If research is what is for you, then look for job in academics or FDA, or that postdoc definitely helps quite a bit in establishing contacts, in understanding the resources that are available to you, and in how to utilize those resources to the fullest. So yes, do that. And if you're here and if you are in a science background and if you want a job in FDA, please let me know.
Yeah,
I'm very
fantastic.
glad to know.
Yeah, fantastic. So Nitya, I have a request to you. Just have a look at our YouTube. There might be some interesting questions. So please,
Yeah, sure, sir.
we'll pick up some questions from there. And before going to that, I have one question about the SNP based sequencing. Now it's it's like a everyday affair. And have you ever taken this, or you can do that FDA, I'm not sure, like 23andMe getting really popular in the US. So what is your take on this, the SNP-based genome sequencing, or at least the genotyping? And would it alleviate the racism to a certain extent, to know that you have blood from, the Africans and Asians and Scandinavians? So it's like a mix. What is your take on this?
I personally have not taken 23andMe, but I have several colleagues, they have taken it because they were so interested in how these things work. And yes, the results, I have seen the results. The results are pretty interesting to say, because you never know, like, a certain person can have Scandinavian origin, or like you said, an African origin. So which which again supports the theory of evolution, right? It's yes. So which again supports that everybody came from, like, through evolution. You just didn't get dropped by God or like, so you are not superior to another person who's standing right next to you. Never. Like, he's superior in his own ways and you're superior in your own way. So to answer your question about racism, that's more of a very complex issue. If it is a science-based issue, we would have answers right now, Felix. I don't think it's a science-based issue at all, the way that people act. As for science-based issue, if it was a science-based issue, yes, it would. These 23andMe, looking at these phylogeny, looking at where your DNA came from, looking at your ancestry, history, and all that stuff will definitely alleviate that. But it's not a science-based question, right? It's a very social, economic, and political based thing. So I don't know how to answer the question. But yes, 23andMe, I truly think that it is true science.
I understand it's a tough question to answer to. Well, ma'am, we have some questions from the YouTube chat here. So many of the viewers, they wish you greetings. And Kishore Joshi says it's an inspirational story. So Shruti has a question. She's asking, ma'am, I would like to know how easy is it to convert dependent visa into H-1B visa, or do you recommend getting a student visa from the beginning for entering into USA?
For entering into USA, yes, get a student visa. But if you come here as a dependent, like I said, definitely look for science-based job opportunities in universities, science-based universities. They will have grants, and the grant can sponsor H-1B. And the science-based opportunities does not fall under the quota. So you have a separate STEM quota, and also the university science-based doesn't fall under any quota. So they can sponsor as much H-1B as they want, as many they want. And they can also expedite the H-1B, depending, saying that the grant starts like, next month. So we do need this expedited. But that is once you're here on a dependent visa and then you're looking for job opportunities. But if you want to enter the US, then I would highly recommend to have a student visa.
Do you have any favorite time
All
management?
right, thank you, ma'am. I hope.
Padmini. So yeah, sorry, Nitya. So I'm just wondering, because you said about the FDA experience. So do you have any favorite time management trick or tactic? So how do you manage your time? Do you schedule? You said about writing, journaling. I'm sure you're doing some journaling. And how do you manage your time? And do you use any workplace communication tool like Slack? Yeah.
Yes, we do Slack, and we do Teams quite a bit. And we do have a lot of virtual. Obviously, we are living in a virtual world during this pandemic, and I'm sure even after this pandemic, people would prefer that rather, because you do utilize the time. But even before the pandemic started, we have FDA in different countries, and especially, and also we have 3 time zones in the US, and within the US, if we want to communicate. So it was a lot of virtual meetings. So we definitely did Slack and internal Skype, where we have task lists that we maintain. And we have something called a SharePoint, where we can share document and we can live edit. So I can edit and the collaborator can edit, and we can see each other the live edit. So all that tools definitely helps, and it kind of helps their thinking also, like in a sense, you're collaborating with somebody, and especially to a policy person, right? They are not from a science background, but you are not from a law background. So when you want to translate science to law, and then the policy people want to understand science to create the law, it's a pretty tedious process. So you have to communicate in a really efficient, succinct way, so that it's pretty clear and precise. So in that case, like when you have these writing documents that you do live edit, like you see their edits live, and then you see, oh, okay, so he really doesn't understand what this is. That's because in my mind, I know what this is, like, so I haven't explained it well. So then you go back and you explain it in layman terms. So I would definitely, just for fun's sake, do a small presentation, do a live edit, like see whether whatever that you're trying to communicate, the other person understands that. And then just write a really simple science experiment, like write it down 10 steps. Ask the other person to actually do it in the lab, and they'd be like, what is this? That's when you would know, yes, I have not explained it well enough, or I've explained certain steps too well that they're confused as to what to do. So then you take a step back. So yeah, those were the efficient tools that helped me in understanding. Thanks.
Yes, Nitya?
Oh, very true, ma'am. Even I'm using such tools these days, and it's become a common tool also for most of the people all over the world. And I do have a personal question from my side. And my heart says that it would be great if you could be part of our mentor and mentee program if you find little time in your schedules.
I would love to. I would definitely love to. So
I will keep you informed about this program and
sure.
how to mentor a couple of students. And yeah, before leaving, I also would like to know a little bit. You started talking a little bit about the communication. So US is something like the world's village. You can see the people from all around the world in the US, especially in a workplace like FDA. I don't know about the how diverse the group in FDA is. But yeah, in the US, yeah, I've been only once in the US. Yeah. So my question to you is that how do you actually deal with this intercultural or cross-cultural communication? Do you have any tips on it? Because you were raised in Tamil Nadu and it's more or less homogeneous population. And now that you're like in the midst of everybody all around the world, right? So how do you manage this kind of communication? Yeah.
That's a great question, Felix. I have to tell you a really small incident. I know we're running out of time, but I'm going to tell you a really small incident. In India, like we always shake the head like this, right? And then we do this, right? So initially, when I joined, my first in a meeting, like we were all sitting in a meeting. It's the second meeting at Hopkins that I'm having, where I am presenting the data. I wanted to agree with the PI, but I'm writing as he is saying, like, okay, so this is the questions that we have. And I'm going like, but I wanted to say, like, this doesn't work. And he got really excited. He's like, oh, Padmini agrees with me. And I go like, oh, am I? And then he says, aren't you? But I couldn't come out and say I'm not agreeing, because I thought that's highly disrespectful. So action wise, I didn't communicate what I wanted to communicate, and verbally, I couldn't communicate what I wanted to communicate. So I was like a little bit taken aback, and I was like, Rich, I know that that's what you said, but I have a really small correction. And then he goes at the end, that's not a small correction, Padmini, you completely disagreed with me. And then I'm like, yes, I did. And then why did you shake your head? And I was like, yeah. So this is a very small incident, but I have been in situations where I have failed to communicate better, because it's different. Because in the US, like when you say that a particular assay is not working or anything, you don't have to go into detail on what are the efforts that you put into it, why it didn't work or anything. So my first presentation was like really long, and people got really bored. Why should I listen to all this? They want like precise scientific aspects on why it didn't work and how you moved on and how you changed the question. So you have to learn from your mistakes, Felix.
Yeah, correct.
That's
Yeah.
what I did. And it also depends on who you are working with. Like if you are working with another person from another country and not from the US, it's a little bit different. And if you are working with the ENPI, it's a little bit different. Because in that case, he would expect a detailed explanation of why you went about this approach. But if you're working with a person from the US who grew up in the US, they don't expect you to like go into every single detail, but they expect the highlight points, like, okay, these are the things that didn't work. So yes, by learning through your mistakes, and by also understanding the surroundings in a setting, like what kind of setting you are in. So yeah.
Yeah, learning from mistake. You highlighted how the science works. Yeah, that is what the scientific method is. Of course, part of the lifestyle. Fantastic. Yeah, I'm really happy. And yeah, you highlighted the high context and low context. The direct communication versus indirect. So we guys here in India or rest of the Asian countries, we are kind of Africans too, kind of indirect. Non-verbal cues a lot, but
Yeah.
Western Europeans or the Canadians and US is more direct communication, isn't it? Very interesting tips. And before leaving, I would like to have one final question to you. I mean, a lot of people are watching right now, and also a large number of people will watch this episode once again because it's archived in our YouTube channel. So, what is your one key message to the scientists of the country? They are all young scientists. So, what is your message to them?
That's a very, very loaded question. I would definitely say trust in science, learn from your mistakes, make as many mistakes as you can, never ever be embarrassed by your mistake. And once you learn from your mistake, try to understand how you made that mistake, and how you can avoid it. Like when I say learn, not just that you shouldn't make it or you shouldn't approach it. No, definitely understand why you made that mistake and what could you have done differently. Because science is all about that, like Felix said. Like science is all about asking the correct question, and if you don't ask the correct question, that's completely fine. Learn about how to ask the correct question. And please trust in science, and please trust in yourself, and pursue science. Science is the answer for everything tomorrow, for climate change, for any disease, for saving the earth, like, biodegradation, like getting rid of pollution. And it eventually makes you a better person. Please understand science and follow science, and please be passionate about science.
Yeah, fantastic. I'm really happy that you highlighted. So we really need evangelists for the science, isn't it? Nowadays, nobody actually cares about the science. And yeah, I mean, amazing. Yeah, you actually said about the importance of the fail better. Of course, learn from the mistake. I really enjoyed that talk. But
Yeah.
and yeah, thanks a lot.
And I also want to say one thing. I know youth are after money, like in the sense that stability and money, science gives you that. So please understand that. Please understand science does give you stability. Science does pay your bills, and science gives you money. Don't get scared.
Yeah, great, great. Thanks a lot, Padmini. I really enjoyed this
Okay.
interaction. And
I did too. I did. Yeah.
and
Thank you so
welcome to
much.
Young Academy of India. Now I induct you as a fellow, so let's work together for communication
Welcome, ma'am.
and spread the
Thank you.
word.
Thank you. Thank you. I'm pretty
Nitya, you
excited.
have any
Thank
word
you.
to say? We are closing this session.
Yes, two sentences, sir. Thank you for sharing the funny story of nodding the head, ma'am. It makes sense too. I hope that all the upcoming scientists get as good as in-laws like you, the supportive in-laws, and I hope they would reach higher heights in the career. Thank you for joining us, ma'am.
Thank you. Thank you so much for having me. I truly, truly enjoyed this. Thank you.
Thank you all, and please take care. And yeah, see you
Yeah.
Stay
again
safe.
next YS